Okay, so I'll start with the introduction, which is that an enzyme is a substrate that catalyzes or speeds up a biochemical reaction, and they are large molecules and are mainly proteins, and they are made up of a chain of amino acids. Enzymes lower the activation energy, which speeds up the reaction, and they do this by binding a reactant to a substrate. Enzymes are important to living organisms because they allow reactions in the body to happen quickly, and if enzymes didn't exist, the reactions wouldn't be able to happen fast enough for a living organism to survive. In our experiment, the enzyme trypsin was supposed to break down the protein azocasin. The trypsin then hydrolyzes the peptide bonds in the protein, which creates smaller peptide bonds and amino acids. The TCA was then added to stop the reaction. In living organisms, trypsin breaks down food in the small intestine, and again, the pH is usually around 8 where the trypsin is active. And the question we were asking in this experiment was what salt concentration would cause trypsin the question we were asking in this experiment was what salt concentration would cause trypsin to denature the most, and in turn causing the amount of protein added to break down the least. And we hypothesized that a salt concentration of 4.8 m would cause the enzyme to denature the most, causing the protein to break down the least at this concentration. If the salt concentration is close to 0 in the pancreas, the charged amino acid sides of the enzyme molecules will attract to each other, and the enzyme will denature and form an inactive precipitate. Okay, so my abstract was that trypsin is a proteolytic enzyme and is formed in the small intestine, and it is found in a pH of about 8 in the small intestine. Trypsin's function is to digest proteins, which is done by catalyzing the hydrolysis of peptide bonds of the amino acids lysine and arginine on the carbonyl side, and this creates smaller amino acids and peptide chains. Trypsin, as well as other enzymes, can be affected by temperature, pH, and salt concentrations. The question we were asking in this experiment was what I mentioned earlier, which is what salt concentrations would cause trypsin to denature the most, and in turn cause the amount of protein added to break down the least. And we hypothesized that a salt concentration of 4.8 m would cause the enzyme to denature the most, causing the protein to break down the least at this concentration. We tested the salt concentrations 0.075 m, 0.3 m, 1.2 m, and 4.8 m at 52 degrees Celsius and at a pH of 9. Our hypothesis wasn't supported because of an error made during the experiment, which caused a large margin of error. However, our results did show a significant correlation between low amounts of salt and the enzyme denaturing. Materials and methods. So the conditions of our experiment were the different salt concentrations, which were 0.075 m, 0.3 m, 1.2 m, and 4.8 m at 52 degrees Celsius and at a pH of 9. And we added TCA to the reaction to stop the breaking down of the protein so we could measure how much of the protein was broken down by the trypsin in the different salt concentrations. We then centrifuged to remove the undigested azocasin and the enzyme which made up the pellet, and we centrifuged so the amount of protein for the different we then centrifuged to remove the undigested azocasin and the enzyme. And then we centrifuged the amount of protein for different salt concentration so that different salt concentrations could be measured. And azocasin was the substrate used for the quantitative assay of the proteolytic enzyme activity. And the sulfonamide groups added are covalently bonded to the protein, giving it its orange color, allowing it to be absorbed at 440 nm. And we chose the temperature of 52 Celsius and a pH of 9 because they are optimum for enzyme activity and they were controls, and we had a negative control for each condition and a replicate for each condition and a negative control, and we did this to make our results as accurate as possible. And we had 4 tubes of each concentration and 4 tubes of each concentration as a negative control, making that 32 replicates in total. So for results, based on our results, our hypothesis that a salt concentration of 4.8 m would cause trypsin to break down the least amount of protein was not supported. Our hypothesis couldn't be supported due to an error made while conducting the lab, making our salt concentration at 4.8 m have a massive error bar, which is right here. This made up our p-value for salt concentration at 1.2 m compared to 4.8 m to not be significant, since there was a 9.1% chance of it happening randomly. However, there was a significant difference between 4.8 m compared to 0.3 m and 0.07 m, where the p-value for both was less than 5% chance of it happening randomly. And this supports that the less amount of salt there is, the more the enzyme would break down, and the averages in the result column represent the amount that the enzyme broke down. Okay, so for discussion, the experimental design was adequate to test the hypothesis. However, because of our errors, the experiment prevented adequate hypothesis testing. So the hypothesis is not supported by the results, and I already said this earlier, but about the 9.1%. And the p-values are significant in most of the differences in the salt concentrations, but not all, like I said in the results. And we used the optimal temperature and pH for trypsin activity in our experiment, which was 52 degrees Celsius and a pH of 9. And this gave us the highest possible enzyme activity so we could get the most accurate results. Some sources of variability are the absorbent of the 4.8 m salt concentration, and we did not change the pipette tips between each of the different 4.8 m samples. This seems to have contaminated the sample's concentration and thus affected the absorbance, and this caused there to be a huge error bar in the analysis and for our hypothesis, which was based on the 4.8 m data, to be inconclusive. And we did not have any replicates in the first experiment. In the second experiment, we had 3 replicates for each concentration and each negative control. And bacteria is best grown in an environment with a slightly basic pH, about 9, a very hot temperature, 52 degrees Celsius, and a low salt concentration between 0.07 m and 0.3 m.